So my research question was, does Pestiloteuthis microspora have the ability to degrade polyethylene? So plastic pollution is a pandemic that is plaguing this world. According to Bardwaj in the Journal of Polymers and the Environment, 4.8 to 12.7 million metric tons of plastic entered the ocean in just 2010. And according to the UN, by 2050, our population will be reaching 10 billion people. Therefore, the demand for plastic will only go up, and thus with that, plastic pollution in the environment. As of now, there are two feasible solutions to clean up this plastic pollution problem. One of those being the physical collection of plastic, either by humans or machines, etc., or through bioremediation. And bioremediation is the use of living organisms to cleanse the environment of certain environmental pollutants, whether that be genetically engineered bacteria cleaning up oil spills, or plants used to detoxify the soil of lead. So some examples of polyethylene: it's a type of plastic, and according to the UN it can be found in shampoo bottles, milk bottles, plastic bags such as the one pictured here. And according to the NOAA, every single piece of plastic ever produced still persists to this day. This is because very few things have the ability to degrade plastic into other compounds, and so it just stays the same compound, just in smaller sizes, into what we call microplastics, which are plastics that are less than 5 millimeters in length. And some of the effects that this has on animals, according to the Center for Biological Diversity, is intestinal injury if they're consumed, and even death. And additionally, what we see with plastics is it biomagnifies as you go up the food chain, therefore eventually becoming very concentrated in animals that we humans eat. So Pestiloteuthis microspora is a type of fungus that has been found to have the ability to degrade polyurethane, which is a type of plastic. And this was found when a study conducted by Andrew Russell in the Journal of Applied Environmental Microbiology, that it can degrade polyurethane using it as its sole carbon source. And it does this by producing an extracellular enzyme within the serine hydrolase family. And it does that so it secretes that enzyme, which then breaks the plastic down into other compounds that can be consumed and utilized by the fungus. Additionally, other microorganisms and fungi have bioremediation capabilities. However, Pestiloteuthis microspora's ability to degrade polyethylene remains untested. So my hypothesis was that PE microspora will be able to degrade polyethylene, and I chose that because it had been found to have the ability to degrade polyurethane, so perhaps it could degrade another type of plastic, polyethylene. So my method was fourfold. So first I had to cultivate the fungus, and this was done using a potato dextrose agar, and that was chosen because that was the same agar used by Andrew Russell to cultivate his fungus. So approximately 12 milliliters of that was poured into each petri dish, left to solidify at 4 degrees Celsius, and then the fungus was applied to these plates. 0.1 milliliters of the fungal stock culture obtained from outgrow was inoculated into each plate, and 4 petri dishes were inoculated. For the experimental culture preparation, so I had to create a medium whereby I could guarantee that the same amount of fungus would be placed onto both the control and experimental plates. And so what we did was use a cell scraper to transfer some of the fungus from a cultivation plate into a PDA broth that was then vortexed and placed into an incubator at 37 degrees Celsius for 4 days. So for the control plate creation, I just used plain PDA plates, and 0.1 milliliters of the vortexed fungal broth that I mentioned previously was inoculated onto each plate. These were then incubated at 37 degrees Celsius for 4 days, and in total around 50 control plates were made throughout the study. Now, the experimental plate creation was slightly different, so I followed a method that was used by Ivano Gruner in the journal PLOS. What he did was, in order to quantify other fungi's ability to degrade polyethylene, he put 10 grams of polyethylene powder into a hot, sterile agar solution just before pouring. This was then thoroughly mixed and 12 milliliters poured per plate, and then 0.1 milliliters of the fungal broth culture was inoculated into each plate. Left for 4 days, incubated at 37 degrees Celsius. Now, how we quantified polyethylene degradation was through what Andrew Russell called the zone of clearance. Now, regular PDA has a translucent appearance. However, when you add a polyethylene to it, it gives it an opaque appearance. And if the fungus had the ability to degrade polyethylene, a zone of clearance would be observed, whereby the opaque agar shown here would turn into a translucent agar here, showing that the polyethylene had indeed been consumed. And in order to clean up, all petri dishes were autoclaved at 121 degrees Celsius for 30 minutes, then disposed of in the conventional garbage. Now for the data. So, unfortunately, no zone of clearance was observed on any of the experimental plates, suggesting that mycelial P. microspora does not have the ability to degrade polyethylene. However, other data was gathered as well, that being how many fungal units or colonies were observed on both the control and the polyethylene plates. And that's shown here by this graph. This is a graph of the mean fungal colonies for the experimental and controlled trials, as well as the error bars representing one standard deviation away from the mean. So in order to see if this data was statistically significant, a t-test was conducted, and the inferential statistics shown here suggested that the control plates had a statistically significantly greater amount of fungal colonies than did the polyethylene plates. And this image down here is an experimental plate. As you can see, it has that opaque appearance, and this had been inoculated and incubated for 4 days, and as you can see, there was no zone of clearance observed. So why did the polyethylene affect the growth of the fungus? Well, one possible explanation could be the displacement of nutrients within the agar. So the added polyethylene caused a decrease in the density of the nutrients because it increased the volume of the solution. And according to Supercrop in the Canadian Journal of Botany, less nutrients or a lower density of nutrients causes less growth in fungi. So that could perhaps explain why there was less fungal colonies. However, further research is necessary to determine if that was the cause. Now, there were some limitations to my experiment, one being the stage of growth that the fungus was utilized in. So, the mycelial stage of growth transitions into the vegetative state of growth for fungi, and the mycelial stage was utilized in this experiment as opposed to the vegetative state of growth in both the Brunner and Russell experiments. The reason why is because in order to grow the fungus to the vegetative state, that outlasted the time constraints of this experiment, therefore the mycelial stage had to be used. And perhaps the reason why the fungus could degrade polyethylene is because the vegetative state is the first time that the serine hydrolase enzyme is produced. We're not sure, and further research is necessary to determine that. Additionally, the agar type was slightly different from the Brunner paper. So, as I mentioned previously in the methods, PDA was used in the control and also in the experimental. But in the Brunner experiment, a custom agar was utilized. And the reason why I didn't use that is because some of the volatility of some of the reagents wasn't suitable for a high school lab, so I had to go with a much safer option. However, according to Lariche, in the American Society for Horticultural Science, PDA has the same nutrients as the custom agar did, but it is likely that the concentrations of these nutrients were dissimilar, and therefore that may have affected the fungus's ability to degrade polyethylene, but further research is necessary. One more limitation would be the density of the fungi in the broth varied. So because I used the fungal broth as a medium for the control and experimental plates, I used that broth over many days, even weeks, and due to that, because the fungus's concentration within that broth would have varied over time as it consumed nutrients, which means that most likely the mean fungal colonies observed in the plates would have varied depending on when they were plated with the age of the broth. This would have caused changes in the standard deviation and the mean. However, every single time, no zone of clearance was observed in the polyethylene plates, regardless of the age of the broth. What are some of the implications of this experiment? Well, one being is that, as I mentioned previously, bioremediation is a possible solution to plastic pollution, and this narrows down the possible candidates that could be used for bioremediation. And fungi are a cheap and feasible option to be used. However, further research is necessary because we have to find out if these fungi or other microorganisms are persistent in an ecosystem, perhaps after they've degraded all plastic, they become an invasive species and mess up the whole ecosystem? Or are they efficient at many different temperatures that may narrow down the latitudes that they can be deployed at? Additionally, what other plastics can be degraded? I only tested one here, but there are many others that are utilized by humans every single day and are thrown away to pollute the environment. And that's it.
Alright. Think about the initial curiosity that led you to your inquiry. What other areas of inquiry might that same curiosity lead to?
Well, I would say that how this first experiment started was I was interested in bioremediation of the environment. And I would say that I would still stay within that realm, but perhaps test other kingdoms of organisms, such as bacteria. I know recently a bacteria in the Mariana Trench was discovered that has the ability to degrade oil and consume that, which could help with oil spills, etc. So maybe you could do some tests on that. Additionally, I did intend at the start to use a bacteria to test its ability to degrade plastic, because there was this bacteria discovered by Japanese researchers, I believe, called Ideonella sakiensis, that can degrade PET, which is a type of polyethylene. But because I couldn't obtain that bacteria, I didn't use it, but I would probably like to investigate that.
Okay, Reese, based on the questions that you presented that could be further researched, what advice would you give other researchers who might choose to investigate the questions?
What advice would I give? Can you repeat the question, please?
Based on the questions that you presented that could be further researched, what advice would you give researchers who would then go and research those questions?
The advice that I would give would be to try and follow the method of whoever you're basing your method off of as closely as possible, because as I mentioned previously, both the fungal stage of growth as well as the agar type, two big errors in my method that were not similar to the Bruner paper that I utilized as the basis for my method. So if you follow that as closely as possible, it narrows down the amount of limitations that your experiment has.
All right, so give us one obstacle or challenge that you encountered while you were going through your research method, and how did you address that obstacle?
Okay. Well, one, as I said previously, was the stage of growth of the fungus, mycelial as opposed to vegetative. Now, I did want to pursue the vegetative stage of growth, however, due to time constraints, we really weren't sure how long that was going to take. There was a method that we did find that would help us get to that stage of growth, however, we weren't sure within the time constraints of the experiment whether that was going to be feasible to reach that in time and then test 100 plates with that. So that's probably what I would do.